Right, thank you very much. I have the unenviable task of being your last big speaker before lunch. But in a way it's good because in Brussels 2 weeks ago I was Rod's warm-up act, and so now he's my warm-up act. So I feel that that's, we're even now, it's a little fairer. So I'm a postdoc at Newcastle University. I did my PhD there and finished in 2012, and I've been working there ever since on various different projects. My PhD was in leukemia biology, so understanding more about what actually causes leukemia in the first place. But I've kind of gone off in several different directions since then. My current research interests are actually more towards childhood cancer survivorship, but sort of molecular biology, i.e. what causes the long-term effects on a molecular level that we sometimes see in survivors. And my current project is neurotoxicity, which is attributed to methotrexate treatment in leukemia. And if any of you read Contact magazine, you may have seen an article I wrote about a year ago, I think, kind of describing the project and hoping I was getting funding for it. So what you're going to see today is, yes, I did get funding for it, and I have been working on it full-time since February. So I'm going to show you a little bit of my data, including some pictures of cells from 2 days ago, because you invite scientists to talk about their work and they want to show you everything. So I hope I don't overrun too much. But my backstory, I think, is kind of unusual. This was me in 1994, just about half a mile down the road at St Bartholomew's Hospital. I was diagnosed with acute lymphoblastic leukemia, B-ALL, and I was given an intermediate prognosis. But here I am in Newcastle, not too long ago, still playing with various liquids, but not with super soakers anymore, although I think that would be a fine idea. Never health and safety would put that through, though. So why do we actually want to do survivorship research? Well, as you can see from this graph here, the survival of, I think it's almost all childhood cancers, is going up. And so we have more childhood cancer survivors in the world, in the UK, in Europe, all over the world, than ever before. So we need to, as well as carrying on researching better treatments, kinder treatments, and of course treatments for those childhood cancers which are still sadly very hard to cure, we need to look at what happens to people from their treatment in the long term. So as you know, there's a variety of treatments used to treat childhood cancer. We've got radiotherapy, we've got chemotherapy, and of course we've got surgery, and then we've got new therapies such as immunotherapy, which you've heard a little bit about this morning. But for the purpose of my talk, I'm going to mainly talk about chemotherapy, because it's what I know most about. So these are just 8 drugs which are used in the treatment of childhood leukemia. They are 8 that I had 21 years ago. They are 8 that children will get now as well. And they are only 8 of many. There are more than this, but I've just put this up to kind of accentuate the fact that although we have come a long way in terms of the treatment of childhood leukemia, so when I had it 21 years ago, I think the overall survival rate was about 70%, and I think we're up to about 90 now. So what we've done is we've kind of juggled these around as when we give them, how much we give them, we're understanding more about the genetic basis of leukemia and how patients are different, and giving them different combinations of these treatments. But with a few exceptions, we have not actually changed the treatments that much. So we need to be looking at the long-term effects of these treatments. And I'm going to talk very briefly about this one here, this bright red one, which I seem to remember made my urine very bright red as well, which I found hilarious at the time, my parents not so much so. I'm going to talk about this one briefly. One of my colleagues in Newcastle is working on it, and then I'm mainly going to talk about methotrexate, which is my main research project. So probably the most well-researched, one of the most well-researched drugs in terms of long-term effects are anthracyclines. So they will be called doxorubicin or daunorubicin or various brand names of these drugs. And essentially, they are really good cancer treatment agents, but we now know that they cause cardiotoxicity through a variety of means. And so my colleague in Newcastle, Chris Plummer, he's a clinical cardiologist, he works on this a lot, and he wanted to know, along with people all around the world working on this, what can we actually do to understand why these drugs actually cause cardiotoxicity, and is there any way that we can prevent this when we've got further understanding? So this is what we would call a late effect. So the signs of this are normally after treatment, and it can be 1 year after treatment or it can be 20 years after treatment. So me having been 21 years in remission now, I'm sort of at peak time for these side effects. So it's really, not something you can just forget about when, a few years after treatment. We're affected long term by these things. So I'm just showing you one of his graphs. So in people who aren't treated with these drugs, this is their general rate of congestive heart failure. So it's about 2.5%, so it's quite low. In people who are treated with a small dose of doxorubicin, so that would be on people who are on maybe intermediate or lighter treatment protocols for leukemia now, the increase goes up to a point and then kind of levels out to be that of a normal population. But for people who are treated with a lot of anthracyclines, so those who have more intensive disease, the risk is quite substantial that they will actually experience heart failure. But look, it's 20 to 30 years after treatment. So we still need these drugs. They're very, at the moment, I hope we don't need them for so much longer because they are very useful and they are good at what they do. But what can we actually do about this? So as Rod mentioned a little bit in his last presentation, we can screen. So I have an echocardiogram every 4 years. I still have echocardiograms every 4 years. And these can pick up some early signs. And if we can pick up early signs of problems, then we can do something about it. Last time I went to my echocardiogram, they essentially dismissed me because I run marathons and half marathons, so I was considered not to particularly be at risk. But I will continue going for my echocardiogram every 4 years. But now there's something that we think we can actually do about it at the time of treatment. So I don't have time to go into them in detail, but there are drugs called ACE inhibitors. And clinical trials and early kind of stage trials are now finding that if we give these to patients during their doxorubicin treatment, this actually protects their heart and doesn't affect the use of the drug against the cancer. So this is kind of a full circle, and we've looked at what biologically this drug does to the heart, and we've found something which we think can actually protect the heart during treatment without affecting the mechanism of the drug. So if you'd like to look up more about them, I suggest you do. So, as I said, my main research project that I've been involved full-time on since February is to do with a drug called methotrexate. And some of you will know about this, obviously, in the scope of childhood leukemia treatment, and I think some other childhood cancers as well. Some of you will know about it in the scope of rheumatoid diseases, such as arthritis. So quite a lot of people are on this drug for arthritis in far smaller doses than we give our children with leukemia. But there are a variety of acute effects. So there's one called stroke-like syndrome, which happens, we think, in about 3 or 4% of patients, where actually the child just gets paralysis. I think it's a day, 2 days normally after one of the treatments, and the child just goes down with either a half-side paralysis, one limb, and seizures as well, severe seizures. And more recently, we found that methotrexate can cause neurocognitive effects. So these are things like visual processing, memory, and unfortunately the recent results seem to suggest that around 50% of survivors will have neurocognitive issues based on this drug. So we don't really know why methotrexate causes these problems, and we don't know for the acute effects why most people don't have these effects and why a few people do. So I was treated on a clinical trial called UK ALL11, just around the corner at Bart's 21 years ago, and I had stroke-like syndrome off one of the methotrexate treatments. So I went home from hospital that night, I think it was. I was playing a board game with my family, and I kept knocking things over. I was really clumsy. And Mum just thought I was playing up and, go to bed, fine, okay. Woke up the next day and I was paralysed down my left-hand side. And I got rushed into hospital, obviously, and after about 2 to 3 days, I think it eventually came back. So I had this personal experience, and I was at an event, something similar to this, maybe 2 years ago now. It was run by Leukemia Lymphoma Research, and I did a talk about my personal experience, and one of the fathers of somebody who had been very recently treated in Newcastle came up to me and essentially said, "My daughter, she had this too." And I was like, "When was that?" He's like, "Just 6 months ago." And I kind of thought, well, hold on a minute. I kind of assumed this was just an issue with the clinical trial. They're getting the drug sorted, figuring out the dosing. But it still happens. And the more I asked clinical colleagues about it, yes, it does still happen. No, we don't know why. And I spoke to my mum about this, and she was like, "God, that was awful. That was the worst thing that happened to you during treatment, because nobody knew whether you were going to be paralysed permanently." At that time, they didn't know that most children regain their movement. Not all. Some people don't regain all of their, they have weaknesses and things. And for some patients, unfortunately, it's much, much worse than that, comas, and can contribute to toxicity-induced death. So my mum essentially told me to figure it out then. And I did, in no uncertain terms. I did explain to her that I'm not a neuroscientist. I was was at the time a leukemia biologist, and she didn't seem to think that was any kind of excuse. So fair enough. And I just wanted to point out, this is a very active area of research. This is a paper that came out last Wednesday from St. Jude's, which is the biggest paediatric treatment centre, I think, in the United States. And this essentially has very conclusively shown that methotrexate is responsible for neurocognitive issues. It's a very, very good paper if anyone would like to have a look at it. Anyway. So I'm going to show you some slides that I show people I present to in the scientific community, because I think we as scientists have a responsibility to present to everyone. But if I don't make any sense, please do pull me up on it later, and I will be happy to explain everything to you, no problem. But essentially, all you need to know about methotrexate is what it does is it depletes folate. So you'll have all heard about B12 and folate being really important in pregnancy. Well, folate is really important because it allows our DNA to copy itself. So if we can reduce the amount of folate that goes into a cell, we can reduce the DNA replicating, meaning we can stop the cells replicating. So that is how methotrexate works. It stops the cell getting something it needs. So that's all well and good, and that's been known for a number of years, really. But what I was more interested in, if I can get my laser pointer working, is this bit of this pathway here. So you can see everything is, all these DHFs and THFs and the 5 ridiculous chemical names are all bits of, like, types of folate in different types. And then there's this process here, which is obviously affected in this pathway, and it has vitamin B12 in it as well. But I was thinking, well, maybe that's got something to do with it. And if you look at what happens on this part of the biochemistry pathway, it goes to produce an amino acid called methionine, which is really important for cells generally, but it's particularly important for this thing called myelin. And myelin is the insulation in all of our nerves. And so if you've heard of the disease multiple sclerosis, which I assume most people have, in that case, what happens is the myelin thins and nerve signals can no longer be sent along the nerve. And so looking at this, I kind of thought, well, hold on. What happens when people appear to have strokes? Nerve signals are no longer being sent properly through the nerves. And so I started talking to a lot of people about this, going, do you think it might be that methotrexate, it has some effect on myelination? And that is the project I'm doing at the moment. Oh, here we go. I've explained it now, but that's what a myelinated neuron looks like. I said that as well. So the question is, what can we do about this? Because we need methotrexate. It's a really useful drug. But when I started speaking to clinical colleagues about other things that could interact to make methotrexate more toxic, we know from research about 20 years ago, if you give methotrexate to rats that have had the anesthetic agent nitrous oxide, it decreases the amount of methotrexate that actually kills them by fourfold. So you only have to give them 25% of the dose that we would normally give them if they've had the anesthetic nitrous oxide. So I said, are we giving nitrous oxide to kids in the UK when they have methotrexate treatment? And my colleagues at Newcastle went, nope, we don't do that. I don't think we'll do that anywhere else. So we surveyed all the paediatric treatment centres in the UK and found half of them still did. I'm proud to report that we no longer do that. We have changed the paediatric treatment protocol for ALL in the UK to ensure that nitrous oxide is never, ever given to children at the same time they have methotrexate, because it's just a risk that we don't want to take. There's other anaesthetics that are available now. Why would we risk that? And there's also other possible drugs that we give the children to manage their symptoms mostly that might be inhibiting this B12 bit here, and the B12 works with the folate in order to do these normal processes. So a small nutritional study in Edinburgh actually said that 25% of patients, just in a very small study, I'd like to point out, are deficient for B12 on treatment, but yet at the moment we don't test patients for their B12 levels before they have the methotrexate. So these are just questions that at the moment, and along with the clinical community, we're trying to figure out, but this is very early, but we have indeed changed the trial protocol. So I did say I was going to put a picture of my cells in, so the way I'm looking at this now is stem cells. So this little blob here, these are cells which actually originally came from foreskin, because that apparently is are the best ones to have, but now they can become anything, and so I'm growing them here, and what I'll do next week probably is I'll put a cocktail of about 8 chemicals on them, and I'm turning them into neurons. So, I know, I mean, I bet that man never thought that his... And there's been hundreds of vials of this man's, and so it's been turned into a remarkable variety of things. But I'm going to turn them into neurons, because the reason for this is children with leukemia have healthy neurons, or they have reasonably healthy neurons. We don't want to be using cancer neuron lines to see how they respond to methotrexate. We want to be using healthy neuron cells. And this is the best thing that science has at the moment, along with potentially mouse models and things which we might consider in the future, to actually investigate this. So if anyone would like to see how these look after I put all the chemicals in, it's going to be about 2.5 months before they turn into neurons and cost X number of thousands of pounds. But you can follow me on Twitter, and I shall post pictures of their progress. So, anyway, I think I'm the third speaker today, or the fourth speaker potentially, that's mentioned Toronto. So Toronto is an incredible place for biomedical research, and more to the point, SickKids, which is their sort of version of our Great Ormond Street, I guess, is an incredible research centre. And what they're doing at the moment is they're doing another study looking at the neurocognitive outcomes of their childhood leukemia survivors. And they think a process called DNA methylation might be responsible. And so I went over there in September and spoke to some of the people doing this, and they're like, oh, you should come back and you should bring some of your neuron cells treated with methotrexate, and we'll look at the same things happening in your model and our patients. And so I went over there, thanks to the European Association for Cancer Research, from April to May this year, and it was absolutely fantastic, and that's the team in Toronto who are doing this work. And so I just want to show you what I've found. So this is DNA methylation. Essentially, the way that our DNA is copied, and our DNA contains blueprints for proteins that are within us and form us, essentially. Methylation is a bit of a weird one. It's not like mutations where bits are missing, so therefore the protein can't be made. It's almost like there's roadblocks. So if you imagine going along a road, cycling along, and then suddenly you hit some traffic cones, you might be able to go around them, you might be able to go straight through them, but they're going to kind of cause a disruption to the way that you handle that. So methylation is basically putting little flags that might slow you down or might disrupt what you're trying to do in that pathway. And so, interestingly with methylation as well is it can be fixed for several years, so 10 years, 20 years potentially. So things that happen to children in treatment might well cause things that happen to them in 20 years' time, where of course things with the blood, generally speaking, it'll recycle itself every couple of weeks, same with your stomach lining. Neural cells, definitely not. Okay, so I treated neural cells in the lab with methotrexate, not the foreskin ones, different ones, and asked the question, does methotrexate actually cause this DNA methylation change? Yes, it does. This is completely pre-published material that I only got about sort of a couple of weeks ago. But if you see these bars here, if there were no changes, they would all be at naught. So you can see with increasing dose of methotrexate, you get an increased number of genes which are increased in methylation. Increases in methylation mean there's less likely to be protein made from that gene. So yes, in brief, methotrexate does cause methylation changes. So what? Well, this experiment I did looks at almost 1 million sites in the genome for methylation changes, and you use various bioinformatics pathways to kind of get your numbers down to a tiny number of genes. You use statistical analyses and algorithms that I can't possibly understand, and I have realized that, and I'm not going to even try. I just speak to the experts. And you get them down into a list which you think is significant, like you think that result is real. And I took nearly 1 million sites of this methylation, and I got it down to just 29 sites, which I thought were important. And one which came up, this is almost like a eureka moment for a scientist, is myelin basic protein. This is the biggest part of myelin. So I take 1 million things, get it down to 30 plausible results, and we find that methotrexate potentially influences myelin. So it's very in keeping with my hypothesis. So changes after one dose of methotrexate are quite small. But if you think that children have 10, 20 intrathecal methotrexates, several high-dose methotrexates, if you get a cumulative change after each one, you might completely alter what happens to those genes. And as I said, this might be fixed long-term, might be. So, very, very quickly, because I know we're going to lunch, I wasn't going to talk about Brexit either. But all I will say is that I have a 15-page protocol, to a 15-page research proposal to carry on this research. In a few years' time, I'm going to Canada in February to work at SickKids for a couple of years. But I'd always planned to come back to the UK and continue this neurotoxicity work. I've already written up a 15-page proposal to be sent to something called the Marie Curie EU Research Fellowship. And I've got experts involved, and it's a really good proposal. If we leave next week, I mothball it. I'm not eligible as a British citizen. And I may be able to do that research at some point in the future, but it won't be the UK. It might be Germany, it might be Denmark, more likely to be Canada, or maybe St. Jude's in the US. So just from a personal, single perspective of what it means to me as a scientist, I mothball a heck of a lot of work if we leave next week. So I think this pretty much supports what a few people earlier said. So what do I see the basic research in survivorship issues? How do I see this going forward, and how do I think we should work together? So more long-term survivors we need more need for research into survivorship issues. And as somebody said earlier, we need to make sure people are living well, not just alive. We need them to have normal, healthy lives as well. And many health conditions that are experienced by survivors, particularly things like mental health, are lacking in basic biological understanding, because we know that survivors also have soaring rates of mental health conditions, for example. And we need to understand the biology of these so that preventions at the time, so like the doxorubicin story, or interventions even later on, like screening for heart problems, can occur. And it's vital also to focus on life-limiting, not just life-threatening survivorship issues. So things like neurocognitive differences, mental health. So again, I think this is almost a carbon copy of one of the slides earlier, so it's good to know we all feel similarly about it. So for the future, we need to ask survivors about their needs and what they want us to research. We need to ask those questions, get the funding to address those questions, whether it be on a societal level, a psychology level, or a basic molecular biology level. And then we need to develop ways of keeping survivors engaged and involved with survivorship research. So I have a 200-person Facebook group that I run at the moment, which is called Childhood Cancer Survivors Unite for Research, I think, and I've been asking them questions that have been proposed here, and people from all over the world are contributing to it, and it's absolutely fantastic. So, and then we also need to collaborate internationally, work with whoever will work with us, wherever they are in the world, to solve these issues and to maximize expertise. And then we need to get any results we find to survivors, helping them as soon as possible. So I'd just like to thank everybody who was involved with this research. As you can see, we work with people all over the country, indeed all over the world, Canada, etc. But I just want to say a special thanks now to somebody who some of you will recognize. And this is Judith Kingston, and she was my consultant at Barts 21 years ago. And more recently, when I started having thoughts about this neurotoxicity work, we met for a coffee just outside Great Ormond Street, actually, about a year and a half ago, maybe, and she had dug out my notes, the masses of my notes. I mean, the poor woman, as you will know, she's very slight, and she, and I said to her, did I have nitrous oxide just before I had my stroke-like syndrome? And about a week before I had my stroke-like syndrome, I had a complication in one of the procedures. It was under nitrous oxide for about sort of 2 hours, which is a lot longer than it would normally be given. So it's not in any way a result, but it's a correlation that helped me develop the hypothesis, and indeed she was incredibly, incredibly supportive and helpful, and it was wonderful to catch up with her and talk to her as a scientist, which I think was weirder for her than it was for me. But yeah, and she sadly passed away, as many of you will know, in I think January, February this year, so she will be missed, and I shall definitely miss her. So thank you very much.